You think you know the ocean, blue water, dolphins leaping, coral gardens in warm shallows, but drop 2,000 meters down and everything changes. The pressure could crush a submarine, the cold bites at 2 degrees Celsius, and in that pitch-black void, creatures evolve that make horror movies look tame. Giant mouths that swallow prey twice their size, eyes bigger than your fist, hooks that spin on tentacles, bioluminescent clouds ejected as weapons. This is not the ocean you learned about in school. This is the deep sea, and every meter down reveals another type of creature so bizarre that scientists had to invent new categories just to classify them. Who are these animals? How do they survive where nothing should live? And what happens when we finally shine a light into the abyss? The Hadal Zone starts at 6,000 meters. That is where the trenches begin. Mariana, Kermadec, Puerto Rico. These are not gentle slopes. These are vertical drops into geology's darkest secrets. The pressure at 6,000 meters hits 800 atmospheres. At the bottom of Challenger Deep, at 10,984 meters, it climbs to 15,750 pounds per square inch. That is 1,000 times the pressure at sea level, the weight of a bull African elephant on every square inch of your body, simultaneously. Nothing with air pockets survives. No swim bladders, no lungs. Everything down there has abandoned the surface world's rules. Anton Frederick Brune proposed the term Hadal in 1956, named for Hades, the Greek god of the underworld. Before that, everyone lumped these depths with the abyssal zone. But the Danish Galathea II expedition in the early 1950s pulled up amphipods from 7,000 meters that looked nothing like abyssal species. The Soviets on the Vityaz found the same thing. A distinct shift in life happens between 6,000 and 7,000 meters. Not a gradual fade, a hard biological boundary. UNESCO now places the official Hadal limit at 6,500 meters. 46 trenches worldwide contain Hadal habitat. Together, they occupy less than 0.25% of the seafloor. But they account for over 40% of the ocean's total depth range. Most of it sits in the Pacific, the largest and deepest conventional ocean division. In 2014, Jeff Drazen and Patty Fryer from the University of Hawaii led the Hadal Ecosystem Studies expedition aboard the research vessel Falkor. They deployed 5 deep sea landers at targeted depths from 5,000 to 10,600 meters. Previous expeditions rushed to the deepest point. Drazen wanted to understand the entire Hadal ecosystem. The team captured the Mariana snailfish at depths between 6,198 and 8,076 meters, Pseudoleparus swirei, a completely unknown variety filmed multiple times during seafloor experiments at 8,143 meters. White, translucent, broad wing like fins, an eel like tail. It glided slowly over the bottom. Alan Jameson from the University of Aberdeen said, "When findings and records can be broken so many times in a single trip, we really get the feeling we are at the frontier of marine science." That was in 2014. The frontier keeps moving. The Mariana snailfish weighs nothing compared to surface fish. 30 centimeters long, top benthic predator in the trench. Feeding on small crustaceans inhabiting the seafloor, its body is tadpole-shaped. Transparent, gooey skin. No scales. The genetic analysis came later. Scientists collected 4 Mariana snailfish from the trench and 4 shallow-dwelling Tanaka's snailfish from the Yellow Sea. DNA sequencing showed the 2 species diverged about 18 million years ago. The Hadal snailfish and other Liparidae species living at around 1,000 meters diverged 9.9 million years ago, right when the Mariana Trench formed. The ancestor adapted to 1,000-meter depths first, then gradually went deeper as the trench opened. The complete absence of light had a profound effect. Substantial loss of vision-related genes. The ability to see no longer mattered. Many circadian rhythm genes vanished, although some essential clock control genes remained. The snailfish kept its rhythmic cycle, just stopped basing it on the sun. 2 genes matter more than anything else. CalDNJ and FTXL27, multiple copies, increased expression. These allow the Hadal snailfish to maintain its auditory senses and withstand pressure that would liquefy most vertebrates. TLDNJ is essential for forming otoliths, structures in the inner ear made of calcium carbonate. Typically, calcium carbonate cannot efficiently accumulate to form otoliths beyond 4,000 meters. The water chemistry changes, but the otoliths of the Hadal snailfish were just as dense as those of Tanaka's snailfish. Multiple gene copies solved a problem that should be unsolvable. Scientists expected TMAO to be the key, trimethylamine N-oxide, a compound that maintains protein function under high pressure. But the expression of the TMAO generating enzyme in Hadal snailfish matched surface-dwelling marine species. Not elevated, the pressure adaptation lies elsewhere. In 2017, A Japanese research crew filmed a new snailfish species at 8,178 meters. Still undescribed. No formal name, just video footage and depth data, the deepest fish ever seen alive, until someone goes deeper. If you value documented deep sea biology and real scientific data like this, subscribe. It is the best way to support our work. Also, comment from which city you are hearing this from. This script is researched and factual, using sources from the Nippon Foundation Nekton Ocean Census published in May 2025, reporting 866 new marine species discoveries, and the Journal of Crustacean Biology, June 2023 issue covering Amphipoda from Hadal depths with specimens from 10,936 meters in Mariana Trench. These are not Wikipedia summaries. These are peer-reviewed publications with specimen counts and depth records. Now back to the information. The amphipods own the Hadal zone. Eric Dahl published the first compilation in 1959, Amphipoda, from depths exceeding 6,000 meters. Since then, amphipods from the superfamilies Lycaenozoidea and Alicelloidea became the best sampled Hadal fauna. They are readily recovered by different sampling techniques: mobile invertebrates, shrimp like crustaceans, sometimes called insects of the sea, typically less than 1 centimeter long, but Hadal amphipods grow 20 times larger, up to 30 centimeters. Hirundellea gigas lives at 10,936 meters in Mariana Trench. Bathycalosoma schellenbergi at 8,370 meters in Puerto Rico Trench. Eurythanes magellanicus at 8,094 meters in Mariana Trench. Stegocephalidae, genus species, at 8,380 meters in Puerto Rico Trench. Over 400 species are known from Hadal ecosystems as of 2020. 83% are recorded from only 1 to 2 Hadal features. High endemic diversity. The trenches are isolated. Each develops its own species. The first in situ images of Hadal amphipods came from the Romanche fracture zone in the Atlantic at 7,500 meters in 1957. 2 photographs. Amphipods active on the seafloor with ophiuroids, anemones, and isopods. A window into a dynamic Hadal community. When researchers place bait on the trench floor with a camera, the imagery is striking. Large aggregations of Hirundellea dubia at 8,000 meters in Kermadec Trench. After 1 hour. After 8 hours, the bait is stripped to bone. The efficiency with which amphipods remove flesh from carrion rivals, surface scavengers. Alicella gigantea, the supergiant, feeding at 5,900 meters in Mariana Trench. Large aggregations of Bathycalosoma schellenbergi in San Cristobal Trench at 7,220 meters. Supergiant amphipods were discovered in 2012 by the University of Aberdeen during an ocean research expedition. No one had observed them since the 1980s, until that expedition pulled up several complete specimens. The benthicicamid prawn, Benthicyximus crenatus, encountered up to 7,703 meters deep in various Pacific trenches. A decapod, crustaceans, including shrimps, lobsters, crabs, crayfish, until researchers discovered Benthicyximus in Kermadec, Japan, and Mariana Trenches in 2009. Decapods were thought absent from the Hadal zone. The oxygen levels seemed too low, but here they were, documented, filmed, collected. Abyssal cuskeel, Abyssobrachula galatheae, may hold the record for deepest fish ever caught. A specimen trawled up from 8,370 meters in Puerto Rico Trench in 1970. The net may not have been fully closed. Possible it was caught shallower. The data remains contested, but the specimen exists, preserved, measured. If the depth is accurate, it beats every snailfish record. The Xenophyophores might be the weirdest, once thought to be sea sponges. Now known to be single-celled organisms with multiple nuclei, capable of growing to the size of a softball, 1 cell, softball-sized. They are fragile. Researchers struggle to retrieve them intact from the ocean deep. They use slime to eat detritus from ocean sediment. Sometimes they colonize bacteria to supplement their diet. They extract materials from the environment to build an exoskeleton called a test. Tests come in wide-ranging shapes and designs. Each offers benefits for filtering and capturing food. They have been photographed on the seafloor at multiple trench sites, sitting there, pulsing gently, a single cell doing the work of an entire organism. The pressure at Hadal depths creates a biological arms race. Every adaptation costs energy. The Hadal grenadier fish, generalist benthic feeders eating crustacean and amphipod prey, they are attracted to hydrothermal vents and hydrocarbon-rich cold seeps. Features prominent in oceanic trenches due to subduction and volcanic activity. The fish patrol these areas, waiting. 8,000 Antarctic toothfish brought aboard by trawlers between 2011 and 2014. 71 showed clear signs of attack by colossal squid. bite marks, hook scars, the squid hunt at depth, the fish barely escape, evidence sits in trawler holds, documented, photographed, measured, now shift up, not Hadal, but still deep. The twilight zone, 600 to 1,000 meters. Then the midnight zone, 1,000 to 4,000 meters. Here, bioluminescence becomes the dominant sense. 90% of deep-sea creatures below 1,500 feet produce their own light. The chemistry is simple. Luciferin, a light-emitting molecule, luciferase, an enzyme. When they combine with oxygen, they release energy as visible light. Most deep-sea creatures produce blue-green light. Blue wavelengths travel farthest in water. Most marine organisms are sensitive to blue light, but some break the rules. The malacostid family, loose jaws, these fish produce red light. They can see it when others cannot. Advantage by seeing prey without making their presence known. The stoplight loose jaw. By creating red light in the deep sea, they see red-colored prey. Communicate, even show prey to other dragonfish. While other unsuspecting animals cannot see their red lights as warning to flee. The dragonfish, 3 separate light organs beneath each eye. 2 shine red lights, night scopes, sneaking up on prey that see only blue lights. The Pacific viperfish, Choliodus macuni, largest in its genus, 30 centimeters long. Iridescent dark silver-blue coloration, overlaid with specialized ultra-black skin, absorbs up to 99.7% of light. Helps control bioluminescent signals. The dual-layer system gives precise control over visibility. Teeth so large the fish has to open its jaws incredibly wide to accommodate them. Ultra-black skin serves dual purpose. Masks the fish's own bioluminescence. Remains hidden from predators and prey. The International Union for Conservation of Nature lists this species as least concern. Despite limited geographic range, their populations remain stable. The deep-sea anglerfish, female Melanostidus johnsoni. Large head, incredibly sharp teeth, a long fishing rod-like structure extending from the top of its head. At the end sits the esca, a ball containing glowing bacteria, Photobacterium. The lure attracts curious prey. Also useful for finding mates in the vast dark expanse. Males are tiny. When a male finds a female, he attaches himself to her body, fusing with her circulatory system, becoming essentially a permanent sperm-producing appendage. Extreme sexual dimorphism. Females can consume prey larger than themselves, expandable stomachs and jaws. Males have enhanced sensory organs to locate females in darkness, a crucial adaptation. After finding a female, the male never lets go. The species is listed as least concern. Widespread distribution across global oceans helps maintain stable populations. Black dragonfish, Ideacanthus atlanticus, the most pronounced sexual dimorphism in deep sea fish. Females reach 35 centimeters. Black body, prominent teeth. Males grow to only 5 centimeters, retain larval features throughout their lives. Females swim at depths less than 500 meters, move closer to surface at night. Males remain in deeper waters between 1,000 and 2,000 meters. The size difference is remarkable. Females are active predators. Nightly vertical migrations suggest they follow prey movements. Males with reduced size and simplified features likely have different feeding strategies. Despite unusual biology, the International Union for Conservation of Nature lists this species as least concern. Widespread distribution across southern oceans helps maintain stable populations. The viperfish hunts with stealth. Photophores line its body. Small light-producing organs. It can turn them on and off, creating patterns. Some species use counter-illumination, producing light on undersides to match downwelling sunlight, becoming invisible to predators below. Others use specific patterns of light pulses to attract mates or warn competitors. Female firefly squid create brilliant blue displays to signal readiness for reproduction, turning deep waters into spectacular light show during breeding seasons. The ostracod crustaceans, sea fireflies. Males release patterns of blue luminescent chemicals into water while swimming in specific trajectories. Each species has unique light signature, ensuring successful reproduction with right partner. The timing and intensity carry additional information: size, health, readiness to reproduce. Female deep-sea dragonfish modulate light patterns to indicate reproductive status. Males respond with distinct signals. 76% of all oceanic marine animals are capable of bioluminescence. They produce their own light through chemical reactions inside their body. Bioluminescence is an active process, can be turned on and off, as opposed to passive traits of fluorescence and phosphorescence. Some bioluminescent organisms generate their own light. Others take up bioluminescent bacteria from water column and house it in light organs. Symbiotic relationship. Marine bioluminescence, commonly expressed as blue-green light. These wavelengths travel further distances through water, more easily visible in deep ocean. Now the cephalopods, the giants, the colossal squid, Mesonychoteuthis hamiltoni, heaviest living invertebrate, 495 kilograms confirmed, perhaps 600 to 700 kilograms based on beaks found in sperm whale stomachs, maximum total length around 4.2 meters. But that excludes the feeding tentacles. In life, the squid's tentacles are hidden, only released when capturing prey. The colossal squid lives in Southern Ocean around Antarctica, circumantarctic distribution, depths from 1,640 feet to over 6,561 feet. Smaller juveniles near surface down to 1,640 feet. Larger juveniles and adults reach depths of 6,561 feet or more. Since 1925, only 9 specimens larger than juveniles have been reported, first seen in natural habitat in March 2025. Most knowledge comes from dead specimens washed ashore or stomach contents of predators, sperm whales. If you value documented deep-sea research and specimen-based science like this, subscribe. It is the best way to support our work. Also, comment from which city you are hearing this from. This script is researched and factual using sources from Wikipedia's Colossal Squid article, updated 2 weeks ago with confirmed mass measurements, and the Natural History Museum's 2025 Giant and Colossal Squid Research, showing first live colossal squid footage from March 2025. These are not speculation pieces. These are database entries with specimen numbers and collection dates. Now back to the information. The colossal squid has largest eyes ever documented, 27 to 30 centimeters in diameter. Some estimates go to 40 centimeters for largest collected specimen. Eyes that size see bioluminescence from vast distances, critical in environment where prey and mates are sparse. The squid is only member of Cranchidae to display hooks on its arms, suckers and tentacles, swiveling hooks, 3-pointed hooks, made of material similar to human fingernails. Perfect for gripping large fish. Other squid, other cephalopods, they combine waste with mucus secreted from suckers to form balls of food. Colossal squid feed primarily on small fish, lanternfish, deep sea smelt, found as stomach contents in adult specimens. They also attack larger fish. Of 8,000 Antarctic toothfish brought aboard by trawlers between 2011 and 2014-71 showed clear signs of attack by colossal squid. A study in Pride's Bay region of Antarctica found squid remains in female colossal squid's stomach. Cannibalism within species. Studies measuring delta-15N content of chitinous beaks demonstrate colossal squid is top predator, positively correlated with increased size. It preys on large fishes and smaller squids according to size. Its predators include sperm whales and sleeper sharks. The giant squid, Architeuthis ducks, longer but lighter. Females measure up to 43 feet from posterior fins to tip of long tentacles, weighing up to 606 pounds. Males slightly shorter at 33 feet. The mantle about 2 meters long. Feeding tentacles concealed in life are 10 meters. Claims of specimens measuring 20 meters or more have not been scientifically documented. Found in all world's oceans, although rarely in tropical or polar regions. Hotspots include North Atlantic off Newfoundland, coast of Southern Africa and South Atlantic, North Pacific off Japan, around New Zealand and Australia in southwestern Pacific. Giant squid inhabit deep ocean, but avoid deep open ocean. They prefer areas with underwater geography, slopes, ridges, seamounts. First photographs of live giant squid in natural habitat came in 2004, off Japan's coast. The squid was feeding. Researchers salvaged a ripped tentacle, confirmed existence. First video emerged in 2016. Since then, sightings increased. Still rare, still difficult. The giant squid has 8 arms and 2 longer tentacles. Arms and tentacles lined with hundreds of subspherical suction cups, 2 to 5 centimeters in diameter, each mounted on stalk, circumference lined with sharp, finely serrated rings of chitin. The perforation of these teeth and suction of cups serve to attach squid to prey. Common to find circular scars from suckers on or close to head of sperm whales that attacked a giant squid. Each tentacular club divided into 3 regions: carpus, manus, dactylus, wrist, hand, finger. The giant squid has sophisticated nervous system, complex brain, largest eyes of any living creature except perhaps colossal squid, up to 27 centimeters in diameter, 9 centimeter pupil. Large eyes better detect light, including bioluminescent light, which is scarce in deep water. The giant squid probably cannot see color, but can discern small differences in tone. Important in low light conditions, giant squid maintain neutral buoyancy through ammonium chloride solution, found throughout their bodies. Lighter than seawater, differs from method used by most fish, which involves gas-filled swim bladder. Analysis of mitochondrial DNA of giant squid individuals from all over the world found little variation between individuals. Just 181 differing genetic base pairs out of 20,331. This suggests only single species of giant squid in world, genetic uniformity across oceans, no regional subspecies, one giant squid, everywhere. Sperm whales hunt both species, circular scars on whale skin from squid suckers, bite marks. Evidence of epic battles in pitch black water. The whales dive to 2,000 meters, sometimes deeper, hold breath for 90 minutes, echolocate squid in darkness, close in, the squid fight back. Hooks pierce whale flesh, suction cups tear skin, but the whales win most encounters. Squid beaks accumulate in whale stomachs, indigestible. Scientists examine these beaks, measure them, estimate squid size. Some beaks suggest colossal squid much larger than any captured specimen, perhaps 700 kilograms, perhaps more. No one knows. The deepest whales hunt the biggest squid. The biggest squid leave the biggest beaks, math that writes itself in whale intestines. Now the vampire squid, Vampiroteuthis infernalis, vampire squid from hell, Latin and Greek combined, small cephalopod found throughout temperate and tropical oceans, extreme deep sea conditions, 600 to 900 meters, sometimes deeper to 3,000 meters, the oxygen minimum zone, OMZ. Within zone saturation of oxygen is too low to support aerobic metabolism in most complex organisms. Vampire squid is only cephalopod able to live entire life cycle in minimum zone at oxygen saturations as low as 3%. That would kill most fish, kill most cephalopods, but vampire squid thrive. To cope with suffocating depths, vampire squids developed several adaptations. Of all deep-sea cephalopods, their mass-specific metabolic rate is lowest. Their blue blood's hemocyanin binds and transports oxygen more efficiently than other cephalopods. Aided by gills with especially large surface area, the animals have weak musculature, greatly reduced shell, but maintain agility and buoyancy with little effort. Sophisticated statocysts, balancing organs akin to humans' inner ear. Ammonium-rich gelatinous tissues, closely matching density of surrounding seawater. The vampire squid's ability to thrive in OMZs also keeps it safe from apex predators that require large amount of oxygen to live. Most predators cannot follow. The vampire squid's large eyes and optic lobes may be adaptation for greater sensitivity to distant bioluminescence, signs of animals, prey aggregations, potential mates. Sensitivity useful when monitoring vast area of water column, which is largely featureless at these depths. The vampire squid lacks ink sacs. If disturbed, it curls arms up outwards and wraps them around body, turning itself inside out, exposing spiny projections. If highly agitated, it ejects a sticky cloud of bioluminescent mucus containing innumerable orbs of blue light from armtips. Not ink, bioluminescent slime. Predators encounter this cloud and hesitate, confused, stunned. The vampire squid escapes in erratic zigzags, using fins to fly through water. Their highly developed staticist contributes to agility. Through video analysis, squid estimated to reach speeds around 2 body lengths per second. Accelerate to these speeds in 5 seconds, not capable of long migrations, not capable of extended fight and flight, but enough to escape immediate threat. Despite name, vampire squid does not feed on blood. It eats marine snow. Mixture of organic debris, dead plankton, and fecal matter drifting down from upper layers. Sedentary generalist feeder, uses 2 retractile filaments to capture food. Filaments have small hairs made of many sensory cells, help detect and secure prey. If filaments contact entity or if vibrations impinge upon them, animals investigate with rapid acrobatic movements. The vampire squid was discovered during Valdivia Expedition 1898 to 1899 led by Carl Chun. Chun was zoologist inspired by Challenger Expedition. He wanted to verify life exists below 300 fathoms, 550 meters. Chun later classified vampire squid into family Vampiroteuthidea. Expedition funded by German Society, Gesellschaft Deutscher Naturforscher und Ärzte. Group of German scientists who believed life existed at depths greater than 550 meters. Contrary to Abyssus theory, Valdivia fitted with equipment for collection of deep-sea organisms, laboratories, specimen jars. Voyage began in Hamburg, followed by Edinburgh, traced around west coast of Africa, navigated around Southern Point, studied deep areas of Indian and Antarctic Ocean. The vampire squid grows to about 28 centimeters. Maximum length? Approximate size of football. Consistency of jellyfish. Most intriguing physical characteristic is proportionally largest eyes of any animal in world relative to body size. Red or blue depending on lighting. Jet black skin. Webbing between arms. Red eyes. Supposedly characteristics of vampire. 8 long arms. 2. Retractile filaments extend well past total length and can be retracted into pockets within web. Filaments function as sensors. Cirri cover entire length. Suckers only on distal half. 2 fins on dorsal surface of mantle. Sexual dimorphism in size. Females, larger than males. First specimens collected in 1903. Originally described as octopus by German teuthologist Carl Chun, later assigned to new order together with several extinct taxa. As phylogenetic relict, it is only known surviving member of order Vampyromorphida. Last of its kind, the gulper eel, pelican eel, Uropharynx, pelechinoids, deep-sea eel. Only known member of genus Uropharynx and family Uropharyngidae. Species belongs to Saccharopharyngiforms. Members historically placed in known order, but now considered true eels in order Anguilliforms. Also referred to as gulper eel, pelican gulper, umbrella mouth gulper. Specific epithet pelechinoids refers to pelican. Fish's large mouth reminiscent of that of pelican. Morphology difficult to describe because specimens so fragile they become damaged when recovered from deep sea's immense pressure. Most knowledge comes from specimens inadvertently caught in deep sea fishing nets. In October 2018, first direct observation of gulper eel made by a group of researchers near Azores. Team witnessed aggressive nature of eel's hunting process, constantly moving around in water column, attempting to find prey. In September 2018, EVI Nautilus team witnessed juvenile gulper eel inflating mouth in attempt to catch prey in Papahanaumokuakea Marine National Monument. Until these explorations, behavior of gulper eels not extensively analyzed. The gulper eel's mouth has capacity to change to inflated shape when hunting, giving mouth massive appearance. Transformation possible due to dual mode, biological morphing mechanism, geometric unfolding of mouth, followed by stretching. When pelican eel in pursuit of prey and opens mouth, the head and jaw structure unfold and spread horizontally, followed by inflation of mouth, inflation made possible given highly stretchable skin of head, characteristic that enables eel to partake in this mechanism, engage in lunge feeding to consume large amounts of prey. When it feeds on prey, water ingested is expelled via gills. Pelican eels, smaller sized eels, grow to about 0.75 meters in length. Lengths of 1 meter plausible. Despite great size of jaws, which occupy about quarter of animal's total length, it has only tiny teeth. Not consistent with regular diet of large fish. Large mouth may be adaptation to allow eel to eat wider variety of prey when food's scarce. Eel can swim into large groups of shrimp or other crustaceans with mouth closed, opening wide as closely approaches prey, scooping them up to be swallowed. Studies show pelican eels are active participants in pursuit of food, not passively waiting for prey to fall into large mouths, hypothesized to exhibit lunge feeding through expansion of mandible and upper jaw. Stomach can stretch and expand to accommodate large meals. Analysis of stomach contents suggests they primarily eat small crustaceans. The pelican eel has long whip-like tail used for movement and communication via bioluminescence. Specimens brought to surface in fishing nets known to have long tails tied into several knots. End of tail bears complex organ with numerous tentacles, glows pink, gives off occasional red flashes, colors displayed through light, emitting photophores, presumably lure to attract prey. Although presence at far end of body from mouth suggests eel may adopt unusual posture to use effectively. Pelican eels black or olive. Some subspecies may have thin lateral white stripe. Coloration especially dark because species exhibits ultra-black camouflage. Special pigmentation reflects less than 0.5% of light, allows eels to be cloaked in darkness in low light environments. Pelican eel found in all world's tropical and temperate oceans, depths ranging from 500 to 6,000 feet, about 150 to 1,800 meters. Researchers from University of Tokyo sequenced mitochondrial DNA from specimens in 2003, found genetic evidence supporting single species, first seen alive near Azores in 2018. Video footage shows eel constantly moving, inflating mouth, attempting to capture prey, aggressive hunting, not passive filter feeding. In 2023, scientists exploring waters near Costa Rica using remotely operated vehicle spotted pelican eel that recently eaten. Belly visibly and dramatically distended. Shape hinted recent dinner was squid. Males undergo change as they mature. Enlargement of olfactory organs, responsible for sense of smell, likely helps them find females. Males also lose tiny teeth around this time. The body diverts all energy into reproduction. Researchers think eels die soon after mating. The deep sea holds more biomass than all rainforests combined, more species than cataloged on land. Most remain undiscovered. In 2024, the Nippon Foundation Nekton Ocean Census announced discovery of 866 new marine species. 10 ocean expeditions, divers, piloted submersibles, remotely operated vehicles, species deemed new to science, guitar-shaped shark, fan-like coral, venomous deep sea snail equipped with harpoon-like teeth, guitar shark found off coast of Mozambique and Tanzania, discovered at depth around 200 meters by David Ebert, also known as Lost Shark Guy, ocean census species, Discovery awardee, only 38th known guitar shark species worldwide. Group SO at risk, 2/3 of species are threatened. Marine gastropod discovered at 200 to 500 meters depths off New Caledonia and Vanuatu by Peter Stahlschmidt from University of Landau, Germany. Turrid gastropods possess venomous harpoons used to catch prey. Produce peptides with potential applications in pain relief and cancer treatment. Drug used to treat chronic pain originally developed from related snail family, underscoring biotechnological promise of new ocean life. In January and February, 2024 international group of scientists, co-led by researcher Ariadna Mech of Barcelona Supercomputing Center, observed 160 species on seamounts off coast of Chile, previously unknown to region. At least 50 suspected new to science, between 50 and 60 potentially new species at first sight, number likely to increase as many samples to work on in laboratory. Also found one of deepest mesophotic corals in world, extending distribution of Polynesian fauna by several hundred kilometers. At depth, found fields of sponges and corals, habitats considered vulnerable and in need of protection, 40-day scientific cruise, unexplored seamounts of Salas y Gomez Ridge, aim to provide critical information to support designation of Salas y Gomez Ridge as ecologically and biologically significant marine area by Convention on Biological Diversity, ecological and socioeconomic priority area for international protection by High Seas Treaty. More than 100 new species discovered on Salas y Gomez and Nazca Ridge, coral and sponge gardens. In November 2025, Senckenberg Ocean Species Alliance published descriptions of 14 new species from ocean depths exceeding 6,000 meters, record-setting mollusk, carnivorous bivalve, popcorn like parasitic isopod. Project aims to make taxonomy faster, more accessible, globally collaborative. One newly described species, Apotectonia senckenbergiae, named in honor of Johanna Rebecca Senckenberg, 1716 to 1743. Naturalist and benefactor whose support for science and medicine helped inspire formation of Senckenberg Society for Nature Research. Amphipod discovered at depth of 2,602 meters in mussel bed within Galapagos Rift hydrothermal vent fields. Some newly identified animals display peculiar traits. Parasitic isopod, Zione. Everta has raised structures on female's back resembling popped corn kernels. Genus, name derived from Ziae, the corn genus, highlights unusual appearance. Found in Australian intertidal zone, represents completely new genus. In July and August 2024, Schmidt Ocean Institute led 28-day expedition, partnership with Ocean Census and Center for Coastal and Ocean Mapping, Joint Hydrographic Center at University of New Hampshire. Researchers created detailed 3D maps of seafloor topography using ship's advanced multi-beam sonar system. Conducted exploratory dive on mountain ridges using remotely operated underwater vehicle. Newly found seamount located about 1,450 kilometers off Chile's coast. Rises 3,100 meters from seafloor. Hosts thriving deep sea ecosystems, ancient corals, glass sponges. One seamount researchers found pristine coral reef covering area of about 800 square meters. Alex David Rogers, Science Director of Ocean Census, said, "Seamounts of southeastern Pacific host remarkable biological diversity with species found nowhere else to date. Scientists estimate they may have found more than 100 species unknown to science during expeditions. Deep-sea corals order Scleractinia, glass sponges class Hexactinellida, sea urchins class Echinoidea, amphipods order Amphipoda, squat lobsters family Galatheidae." Researchers also spotted several rare species, captured first-ever footage of live Promicoteuthis squid, genus so elusive, only 3 species described, mostly from dead specimens collected in late 1800s, Bathyphysa canifera, flying spaghetti monster, documented on unnamed and unexplored seamount along Nazca Ridge. Casper octopus, filmed multiple times, first recording of rare enigmatic squid in natural habitat. Benthic comb jelly, Chelphiella aaftristoma, found on NOAA, ocean exploration expedition in 2018. First time animal appeared nearly identical to Chelphiella. Tristoma had been sampled since 1928. Nicholas Bezio and Alan Collins investigated how new discoveries linked to original species and other comb jellies. Only 2 known species of Platictons benthic comb jellies live in deep sea. One is T. tristoma, deep-sea footage and samples from NOAA. Ocean exploration expeditions suggest many more still undescribed. Specimens collected in 1928 not preserved in a way allowing genetic work. Remains unclear whether either new discovery is T. tristoma itself or just close relatives. Findings show unique genus of deep-sea comb jelly likely plays role in far more deep ocean ecosystems than known before. New lead in genetic data will help future science. Scientific progress takes teamwork, commitment to building on efforts of those who came before, research impossible without samples painstakingly cared for by Smithsonian, modern research conducted by NOAA ocean exploration teams, records created century ago by experts at Natural History Museum of Denmark. Trusting work would lead to future discoveries. Ocean covers 71% of planet, fundamental to life on Earth. Vast biodiversity helps regulate climate, supports health, livelihoods, food security of millions. Much of ocean remains unexplored. Only around 10% of marine species documented thus far. Unlocking mysteries integral to understanding how to protect biodiversity, build climate resilience, secure a sustainable future. 26% of ocean floor mapped to high resolution, working towards full map of seafloor. Would be first time in human history we know what our planet's shape actually is. Findings incredibly important for understanding ocean life in bathypelagic zone. It lies between 1,000 and 4,000 meters deep, largest animal habitat on Earth. In March 2025, a study published in journal Cell analyzed DNA of 11 fishes, snailfish, cusk eels, lizardfish living in Hadal Zone, region about 19,700 feet, 6,000 meters deep and below, to better understand how they evolved under extreme conditions. Researchers used crewed submarines and remotely operated vehicles, collected samples from 3,900 to 25,300 feet, 1,200 to 7,700 meters below water surface, Mariana Trench in Pacific, other trenches in Indian Ocean. Tracing evolution of deep sea fishes, analysis revealed 8 lineages entered deep sea environment at different times. Earliest likely entered deep sea in early Cretaceous period about 145 million years ago. Others reached it during Paleogene 66 million to 23 million years ago. Some species as recently as Neogene period 23 million to 2.6 million years ago. Harmful chemicals used in electrical equipment and appliances until banned in 1970s. Polychlorinated biphenyls, contaminated liver tissues of Hadal snailfish, high concentrations of PCBs and polybrominated diphenyl ethers, flame retardant chemicals used in consumer products until fell out of popularity in early 2000s. Found in sediment cores extracted from more than 32,800 feet to 10,000 meters deep in Mariana Trench. Chemical pollutants in Mariana Trench, microplastics in deep sea. New findings reveal impacts of human activity even in ecosystem far removed from human life. The Mariana Trench now understood to comprise 5 isolated areas, each potentially harboring unique life forms. Discovery coupled with realization, Hadal Zone extends beyond trenches to encompass much wider area. Underscores need for continued exploration in extreme environment. Understanding Mariana Trench's intricate ecosystems, crucial not only for discovering new species. Understanding biodiversity, also for responding to human impacts. Climate change, pollution, deep sea mining. 300 years ago, sailors told stories of sea monsters, kraken wrapping tentacles around ships, dragging them to depths. Those were dismissed as myths, drunken tales, hallucinations. Then, in 1873, a giant squid washed ashore in Newfoundland, 10 meters long, real, measurable, undeniable. The stories were not myths. They were sightings of animals that should not exist, could not exist, by every rule of biology and physics. But they did. They do. Every deep sea expedition finds something new, something impossible, something that rewrites what we thought we knew. In soil cores from Hadal depths, microbiologists found bacteria processing chemicals at pressures that should denature every protein, should collapse every cell membrane, but there they were, metabolizing, reproducing, thriving. The bacteria use different amino acids, different lipids, different everything. Evolution did not run into a wall at 6,000 meters. It climbed the wall, built a house on top, planted a garden. The deepest trenches on Earth are not dead zones. They are biodiversity hotspots, islands of specialization. Each trench hosts species found nowhere else, separated by thousands of kilometers of abyssal plain. No gene flow between populations, each evolving independently. For millions of years, creating a planetary experiment in parallel evolution. What works in Mariana Trench might look nothing like what works in Kermadec Trench. Different solutions to identical problems. That is the power of isolation. That is the terror of the deep. Every creature discussed has one thing in common. They live where they should be impossible. The pressure alone should crush them. The cold should freeze them solid. The darkness should starve them. The oxygen levels should suffocate them. But instead of dying, they dominate. They own those depths. They have turned every disadvantage into an advantage. Pressure that crushes submarines powers their biochemistry. Cold that kills surface fish slows their metabolism to sustainable levels. Darkness that blinds becomes their hunting ground. Oxygen depletion that suffocates most animals becomes their fortress. Protected from predators that need higher oxygen, the deep sea is not a place life tolerates. It is a place life has conquered. And we have barely started looking. The Ocean Census, launched in 2023, 10-year project, goal to discover 100,000 new marine species. As of May 2025, they announced 866, less than 1% of target. In less than 2 years, the rate of discovery is accelerating, not slowing. Every submersible dive finds something. Every trawl net pulls up unknowns. Every sediment core reveals bacteria no one predicted. The deep ocean is not giving up its secrets. It is drowning us in them. Too many species to catalog, too many adaptations to study, too many ecosystems to protect before we even finish mapping them. Climate change is pushing into the abyss. Surface water is warming. Animals migrating deeper, chasing the cold. But the deep sea is not infinite. The trenches have bottoms. The seamounts have limits. Species adapted to oxygen minimum zones are already at lowest oxygen levels possible. If those zones expand upward and downward, squeezing the habitable layer, what happens to animals that cannot migrate, that have nowhere left to go? No one knows. The experiments are being run right now, without controls, without oversight, with stakes measured in extinctions of species we have not even discovered yet. Deep-sea mining companies have applied for permits to scrape the seafloor for rare earth metals, for cobalt, for manganese nodules that took millions of years to form. The nodules are habitat. Xenophyophores grow on them, sessile organisms attach to them. Removing the nodules removes the only hard substrate in areas that are otherwise endless mud. The companies promise to minimize impact, but no one knows what minimal impact means when we do not know what lives there, cannot measure what we might lose. The mining code is being written at International Seabed Authority meetings right now. While you are reading this, decisions made in conference rooms in Kingston, Jamaica will determine fate of Hadal ecosystems no human has ever seen, ever studied, ever valued, until it is too late to save them. But here is what we do know. The Hadal snailfish has eyes that work at 8,000 meters. The colossal squid has hooks that can grip and tear. The vampire squid has blue blood that binds oxygen at 3% saturation. The pelican eel has a mouth that can swallow prey twice its size. The amphipods can strip a fish carcass to bone in 8 hours. The xenophyophores are single cells the size of softballs. These are not accidents, not random flukes of evolution. These are precision-engineered solutions to problems most scientists said were unsolvable, and they are sitting in the deepest parts of the ocean, waiting. For us to notice, for us to study, for us to protect. In 2023, scientists analyzing sediment from 10,000 meters in Mariana Trench found plastic fibers. In 2024, they found industrial chemicals banned in 1970s. In 2025, they found microplastics in amphipod tissues. Nothing is safe. Nowhere is untouched. The deep sea is not remote. It is downstream from everything, every river, every coast, every ship, every factory. What we put in the water ends up in the trench, eventually, always. And the animals cannot leave. They are adapted to pressure, to darkness, to cold. They cannot swim to cleaner water, cannot migrate to safer depths, cannot evolve fast enough to detoxify chemicals they never encountered in evolutionary history. They are trapped in the deepest parts of the ocean, with nowhere to go, and we are filling their world with our waste, our pollution, our noise, our light, our heat, our everything. The first footage of a living colossal squid in its natural habitat was captured in March 2025, 150 years after first specimen found in sperm whale stomach, 150 years of searching, 150 years of wondering, and finally we saw it, moving through pitch black water, tentacles extended, hooks deployed, hunting. The animal was exactly as strange as predicted, and nothing like anyone imagined. Because seeing it alive is different from seeing it dead. Seeing it hunt is different from seeing it measured on a dock. The squid moved with purpose, with intent, with intelligence, not like a mindless predator, like an apex predator that knows exactly what it is doing, that has perfected its craft over millions of years, that owns its environment. That is the deep sea, owned by creatures we are only beginning to understand. Creatures we may lose before we finish cataloging them. before we decode their adaptations, before we learn what they can teach us about pressure and oxygen and survival and evolution and persistence, about living where nothing should live, about thriving where nothing should thrive, about being impossible, and doing it anyway,